Good afternoon, and welcome to the 7th Annual 3-Minute Thesis Competition at Mississippi State University. I'm Dr. Brian Henry, Associate Dean of the Mississippi State University Graduate School. Even though I would rather be speaking to you from Folks Auditorium on our beautiful MSU campus, we're glad you could join us virtually for this exciting display of graduate student research. 3MT is a competition that challenges graduate students to present a compelling verbal presentation of their thesis or dissertation topic and its significance in just 3 minutes, and was developed by the University of Queensland, Australia in 2008. Now, over 900 universities across more than 85 countries worldwide participate in the 3MT. 3MT develops academic, presentation, and research communication skills and supports the development of graduate students' capacity to explain their research effectively to a public audience of individuals who are not specialists in their research area. However, 3MT is not an exercise in trivializing or dumbing down research. It forces students to consolidate their ideas and crystallize their research discoveries. By taking part in the MSU 3MT, graduate students have the opportunity to sharpen their communication skills, promote their field of research to the campus and the general public, and help promote the importance of research and how it positively impacts lives. Winners of this final round will be provided cash prizes including Grand Champion at $1,000, Grand Champion Runner-Up at $750, and People's Choice Award at $750. In a few moments, you will be able to watch the 8 finalists chosen from all submissions that we received. We would like to thank our 11 preliminary judges who reviewed the submissions and chose these finalists to compete in the final competition. Dr. Deborah Lee from the Mitchell Memorial Library, Mr. Terry Kemp, Starkville Utilities Manager, Mr. Les Potts, Finance and Administration, MSU, Mr. Brian Locke, Center for American Vets Director, Mr. Kai Mamaya, HCDC, Mr. Anthony Craven, WMSV Campus Radio, Mr. Michael Richardson, Alumni Association, MSU, Dr. Jason Keith, Bagley College of Engineering, Dr. Donna Gordon, Biological Sciences, Mr. Erdogan Mimili, Ag and Biological Sciences, and Mr. Josh Wooten of TVA. Additionally, I would like to welcome our final competition judges. Kyle Stewart, Executive Director of External Affairs, MSU President's Office. Dr. Eddie Pazant, Superintendent, Starkville-Oktibbeha School District. Hunter Harrington, Director of Membership Development, Starkville Partnership. Hamp Beatty, Starkville Alderman, Ward 5, and Extension Associate in the Extension Center for Government and Community Development. Fran Brock, Extension Agent, MSU Extension Center in Oktibbeha County. Thank you all for volunteering your time to serve as judges to evaluate our final contestants for this exciting competition. We are so grateful for your support of graduate education and development. We know that you do not have an easy choice to make because these 8 presentations are all excellent. This leads me straight into our first presenter. She is a PhD student in the College of Education. Her 3MT presentation is titled, Impact of Prolonged Exposure to Slippery Surfaces on Static Postural Stability. Category of Life and Biomedical Sciences and Engineering. Please welcome Sachini Arachije.
The number of falls that occur at the workplace is increasing each year. These falls can cause significant injuries to the workers, especially life-threatening injuries. One of the main reasons for these falls is loss of balance. In this research, I have studied 2 main factors that could affect somebody's balance: overexertion and nature of the walking surface. More specifically, I studied the changes to the participants' balance over time on a dry surface and on a slippery surface. Speaking of the slippery surface, All the studies that were done so far was focused on short-term exposure to slippery surface, such as 5 minutes or 10 minutes. But if we consider some occupations, such as kitchen staff, military, firefighters, construction workers, they require to walk on slippery surfaces for long hours. So this is the first study that's looking at the impact of prolonged exposure to slippery surface on somebody's balance. For that, my participants had to walk on a slippery surface for 1 hour and on a dry surface for 1 hour, and their balance was tested just before they start walking, at the middle of their walking, and at the end of their walking. That means 0 minutes, 30 minutes, and 60 minutes. The results showed that the participants' balance was significantly decreased when they get to 30 minutes and 60 minutes compared to 0 minutes, regardless of the surface. When we compare the two surfaces, the participants' balance was more decreased when they walk on the slippery surface. In conclusion, participants' balance was decreased over time, and it was more pronounced when they walk on the slippery surface. That balance decrement will make them more fall prone. So what's the importance of this study? As I mentioned, some workers require prolonged exposure to slippery surface. Now we know if they continue to walk, their balance will get decreased. If we could provide them frequent rest intervals, they could work, rest, recover their balance, and start fresh for the next working session. By doing that, their balance will be less impacted. Previous studies have shown even a short break, like 10 minutes, is sufficient for somebody to recover their balance. By doing that, we can minimize the number of occupational falls, which help to preserve the workers' health, improve the workplace productivity, and decrease the amount of money we spend on medical treatment and compensations. Thank you very much.
Our second presenter is a PhD student in the College of Veterinary Medicine. Her presentation is titled Halogenated Anesthetics, Loaded Lipid Emulsions Preconditioning Effect in Canine Primary Hepatocytes In Vitro Study. The category is Life and Biomedical Sciences and Engineering. Please welcome Patricia Elrich.
Hello, my name is Patricia Erich. I'm a PhD student and I'm going to summarize quickly what my project is about in 3 minutes. Since the anesthesia was first developed in the 17th century, it was delivered through the airways, which means that the patient needed to inhale a certain hypnotic or anesthetic agent in order to sleep and stop feeling sensations, so that the surgeon could operate and the patient wouldn't bother him by screaming or crying in pain. In veterinary, it was not different. In fact, we can anesthetize the majority of mammals, reptiles, and birds just by giving them a volatile anesthetic and, of course, with other analgesics and relaxant drugs that will help the anesthesia effect so that the patient can wake up after surgery, ideally like from a beautiful dream, painless and comfortable. In this matter, isoflurane and sevoflurane, both halogenated anesthetics, have been used since the 80s and 90s. But unfortunately, some disadvantages started to pop up, like hypotension and hypothermia, which we don't want our patients to have. But thankfully, some great advantages like anti-inflammatory and organ protection effects began to be an appealing field of research on these two anesthetic agents. Not only these incredible advantages, but also the fact that they could be delivered intravenously or in other routes rather than inhalation. That's when our project comes in, where we planted canine liver cells in 96 well plates and developed an emulsion containing basically water, surfactant, lipoid, and the anesthetic in the liquid form. In order to evaluate the viability and apoptosis responses of these hepatocytes, we added the emulsions at different volumes to mimic what we call an intravenous continuous rate infusion that would be necessary to keep a patient anesthetized for 1, 2, or 4 hours, for example. In addition to that outcome, we agreed on the hypothesis that the emulsified anesthetics were capable of protecting the cells that were submitted to 2 hours of hypoxia plus 24 hours of reoxygenation. And our results incredibly certified that these emulsions were able to decrease the apoptotic response in those hepatocytes in comparison to the lipoid alone. This gave us a great satisfaction on the emotions we developed and on the results that certainly promise a therapeutic effect of these beautiful agents rather than only putting our patients to sleep. For future studies, we plan on administering the same emotions in rats and evaluating specific enzymes from organs like the liver, kidneys, and heart in comparison to control. Thank you very much for listening and lots of science for all of us.
Our third presenter is a PhD student in the Bagley College of Engineering. Her presentation is titled Virtual Design of Hydrokinetic Farm. The category is Physical, Mathematical, Computational Sciences and Engineering. Please welcome Wamina Fajri.
The most prominent renewable energy resource in the US are wind, solar, hydropower, and biofuels. Marine and hydrokinetic resources remain largely untapped and are more reliable compared to wind and solar energy. Mississippi's energy portfolio includes only 3% of renewable energy sources, yet it holds almost 50% of the nation's hydrokinetic power potential. Therefore, the development of this resource is of a strategic importance both for Mississippi and the nation. When you take a look at images of hydrokinetic farms or wind farms, for example, don't you wonder, how can you decide of the positions of the turbines? Is it arbitrary? Should you align them? What is the factor that decides of their position? The technical factor that dictates the placement of the turbines is the wake recovery length. It is the length of the low velocity region located downstream of the turbine. Hydrokinetic turbines extract momentum from the high velocity incoming fluid and generate the wake with the low velocity. If you take a look at these operating turbines in real life, the low velocity region is not visible to the human eye because water is transparent. The same for wind turbines and air. So, how can you investigate a change in velocity and other characteristics if you cannot see them? This is where computational fluid dynamics, or CFD, plays an important role and offers many advantages to flow visualization. My research focuses on using CFD to understand the wake recovery mechanism to build high-efficient turbine designs. The research builds on the hypothesis that the vertical structures that are generated from the turbine blades and the associated burst of high turbulence play a key role in the wake recovery. Therefore, the designs and devices that accelerate the breakdown of these vertical structures and increase the mixing between the surrounding free stream and the wake low velocity can help reduce the wake recovery length, thereby allowing an efficient placement of the turbines in an array. Thank you.
Our fourth presenter is a master's student in the College of Ag and Life Sciences. Her presentation is titled Improving Cotton, the Fabric of Your Life. Her category is Life and Biomedical Sciences and Engineering. Please welcome Grace Fuller.
Hello, my name is Grace Fuller, and I'm a master's student in the Plant and Soil Science Department here at Mississippi State University, and this is my 3-minute thesis. So my thesis is all about improving cotton, the fabric of your life. Now you may be asking yourself, why do we need to improve cotton? Why does it matter? Well, cotton is a plant that produces a fiber that's used in all sorts of products, such as oils, cosmetics, textiles that make our clothing and our towels and things of that nature, and it's used to make money. It's also produced in a lot of big countries around the world, including India, China, and the United States. So it's a really big player in our global economy. Currently, we are facing some problems in cotton production. With the overuse and over-reliance on chemical control techniques such as glyphosate, we've seen an increase in herbicide-resistant weeds. One especially problematic weed is Palmer amaranth. Competitive Palmer amaranth can affect cotton by reducing fiber quality, reducing cotton yield, increasing overhead costs, reducing watering efficiency, and providing a habitat for pests such as rodents and bugs. These problems really emphasize the need for the development of alternative weed management techniques and tools. And one of those possible tools is that of allelopathic crops. Now you may be asking yourself, what is allelopathy? Allelopathy is when a plant produces chemicals, in this case through its roots, that affect other plants. What we're interested in is allelochemicals that make it harder for other plants to grow around the allelopathic plant. We can think of these allelochemicals almost as a bioherbicide. So I'm working with a special variety of cotton known as chromosome substitution or CS cotton lines. And these lines are special because they contain wild or alternative chromosomes from different cotton species that we don't usually cultivate. So these lines have been observed to have some really beneficial characteristics. They have improved fiber quality, they have some herbicide tolerance, and some allelopathic characteristics. So in my research, I tested some previously screened CS lines in the greenhouse using a stair-step technique, and I determined their effects on Palmer amaranth height and chlorophyll reduction. I was able to identify 4 CS lines that reduced Palmer amaranth height and reduced Palmer amaranth chlorophyll concentration. I'm now testing these 4 competitive lines in a field in Pontotoc, Mississippi, and a field here in Starkville to confirm these characteristics or these findings. The next phase of my research is to identify some allelopathic chemicals in the lab and hopefully to identify the gene or genes that are responsible for these characteristics. And this is super exciting because such a discovery would allow these plants to be entered into cotton breeding programs, where eventually this technology would become available to our farmers on the front lines who are battling herbicide-resistant weeds, and ultimately so that the touch and feel of cotton can continue to be the fabric of our lives. Thank you.
Our 5th presenter is a PhD student in the College of Ag and Life Sciences. Her presentation is titled Dietary Selenium Deficiency and Reshaped Gut Bacteria Alter Insulin Signaling in Mice. The category is Life and Biomedical Sciences and Engineering. Please welcome Vicki Huang.
Selenium is a trace element found in Earth's crust. It is observed by plants from the soil and makes its way into animals both on land and in the sea. These organisms then make their way into our local markets where they are consumed by us. Thus, this is how selenium that began in the soil ends up inside us. Concentration of selenium vary greatly between different regions of the world. So, if you live in a big city like New York, you can get foods from different regions, and you can easily make your daily recommended selenium intake obtainable. However, some people living in other areas with lower soil selenium, and if they consume selenium only from local foods, they will be at a much higher risk for selenium deficiency. Nowadays, evidence shows that individuals with suboptimal body selenium status may be prone to certain chronic diseases, such as type 2 diabetes later on in life. National Health and Nutrition Examination Survey also estimated that the mortality rate for type 2 diabetes is increased when your body selenium concentration is lower than 106 microgram per liter. This data also suggests that 10% or about 15.5 million Americans aged 40 years or older have suboptimal body selenium status. In our previous mice study, compared to the mice fed with selenium adequate diet, we found that selenium deficient mice showed type 2 diabetes symptoms, and the abundance of their certain gut bacteria also changed. One of the gut bacteria called Akkermansia mucinifera is significantly increased. Let's call it AM here. AM is a mucin-degrading bacteria which is reported to be a good protector against obesity and type 2 diabetes. In this study, we want to determine the impact of selenium interacts with AM on diabetic mice. Our preliminary data show that selenium deficient mice, after they received AM treatment, you can see the green line here, can improve glucose intolerance and insulin insensitivity. These are the two major indicators of type 2 diabetes. Our future work will focus on the mechanism of how AM prevent type 2 diabetes in selenium deficient mice. The work proposed is also expected to pave the way for research on diabetes prevention by optimizing selenium intake within diets.
Our sixth presenter is a master's student in the College of Arts and Sciences. Her presentation is titled Saving Baby Shark. The category is Physical, Mathematical, Computational Sciences, and Engineering. Please welcome Lindsay Mullins.
Did you know that approximately 4 in 10 Americans are scared to swim in the ocean because of sharks? So I figure, why don't we do all of them a favor and imagine what it would be like to swim in an ocean without sharks? We can kick back, relax, float, and not have to worry about any possible nibbles. Wait a second. Did you feel that on your foot? It was slimy. Oh, it looks like it's just some of that algae that's now covering the ocean floor since sharks are no longer around to keep those seagrass-eating predators in check. We should still try to relax and enjoy the water and, oh, one more thing, those scallops and oysters you're gonna enjoy back at the beach house after this, yeah, unfortunately those are gone now as former shark prey are having a field day on what was gonna be your lunch. But we'll still try to relax and, oh, one last thing. That former shark prey I keep talking about, well, unfortunately, they're sort of depleting their own resources and ruining their habitat from the bottom up. So don't imagine they'll be doing well for too much longer either. In fact, without the balancing role that sharks play, our whole oceans are out of whack. All right, you say, I see how sharks are important. Maybe we can keep them in the oceans. Well, here's some bad news that isn't imaginary. Humans are killing 100 million sharks a year. Does it sound like a lot? Well, it is, and those scenarios we just imagined are very real possibilities. So how can we stop this? Well, sharks are a particular risk in nearshore coastal environments where many species live and where many baby sharks go to reach maturity, but also where habitat degradation and fishing threats are heightened. In order to properly protect sharks, we have to know exactly where these species and baby sharks are and why, and properly protect the habitats that align with those locations and traits. For my thesis, I took a suspected nursery area and known shark fishing hotspot off the coast of Florida and did just that. Using technology, I was able to evaluate and map all of the unique distributions for all of the shark species and baby sharks in the area, as well as identify and quantify environmental parameters such as water temperature or bottom type that may be driving these distributions. In addition, I also found that areas protected from motorized boating activity were associated with higher levels of shark diversity and abundance. With this information, we can explicitly delineate their most vulnerable habitat, identify and protect nursery areas, expand effective conservation measures, and plan for and anticipate changing distributions in the face of climate change. Using resources like these I created in my thesis, we can better manage our shark fisheries, so never again will we have to imagine an ocean without sharks. Thank you.
Our seventh presenter is a master's student in the College of Arts and Sciences. Her presentation is titled A Digital Bioarchaeology, the application of photogrammetry for the recording and analysis of cranial depression fractures from the Mississippi State Asylum. The category is Social and Behavioral Sciences. Please welcome Carrie Porter.
The digital realm has forever changed the ways in which we interact with the world around us, but can it also change the ways in which we learn about people from the past? I'm a bioarchaeologist, and my research seeks to answer this question by using 3D digital technologies to create and analyze digital models of human skeletal remains. Specifically, I use photogrammetry, or the process of creating 3D digital models from 2D images. To create these models, photogrammetric software uses something called feature mapping to analyze digital photographs of an object. This works by recognizing shared features of the object between the multiple images from multiple different angles. The software then places a point at this shared feature and uses spatial information from the photographs to put that point within a 3D space. This process occurs hundreds to thousands of times until eventually a 3D model is formed. Compared to other forms of digital modeling like CT scanning or 3D laser scanning, photogrammetry is portable, it's user-friendly, and it's extremely cost-effective as you basically only need access to a computer and a camera. I use photogrammetry as a method for recording cranial depression fractures, or fractures of the cranial bone that are created by blunt force trauma to the skull. These fractures are great for modeling, not only because they occur on the external surface of the bone, but because they're thought to have high interpretive power when it comes to reconstructing lived experience and interpersonal interactions in the past. This all stems from the concept of the hat brim rule, or the rule that says that cranial injuries that occur above where the brim of a hat would fall are more likely associated with violent conflict than incidental injuries. Previous studies have shown that the individuals buried at the Mississippi State Asylum Cemetery have a high frequency of these fractures, which makes this an ideal collection for asking the question, is photogrammetry an accurate method for the recording and analysis of cranial depression fractures? To answer this question, I'm creating digital models for every individual from the asylum that shows evidence of cranial depression fractures. I then compare those digital models to the fractures that I see on dry bone. Overall, this research intends to provide more information about photogrammetry as a method for recording skeletal remains, but also increase accessibility and application of this technology within bioarchaeology. This can be beneficial to bioarchaeologists in many ways, but particularly for the long-term curation of skeletal collections that may not be forever present, whether that be from poor preservation or from repatriation. Additionally, digital collections can help facilitate international collaboration because they make these collections available to researchers independent of their location, which is particularly poignant with the COVID-19 pandemic. Furthermore, my research contributes to the digital curation of the skeletal remains from the Mississippi State Asylum, and this will help digitally preserve these remains for researchers in the future.
Our eighth presenter is a PhD student in the College of Veterinary Medicine. Her presentation is titled Tipping the Lung Immune Balance. The category is Life and Biomedical Sciences and Engineering. Please welcome Brittany Zaffron.
This past December, a cluster of pneumonia cases in Wuhan, China, led to what we now know as the COVID-19 worldwide pandemic. COVID infections are quickly approaching 50 million, with over 1 million total deaths. The United States leads the world by number of COVID cases. Individuals at risk for severe COVID have diseases such as heart disease, diabetes, asthma, and COPD. However, some young and healthy individuals have died of COVID, suggesting there may be another risk factor we are missing. What if our environment and the chemicals we are exposed to on a daily basis contribute to this risk? This is where my research comes into play. I study the immune effects of toxicants such as pesticides on the lung. Our lungs play an important role in gas exchange and can be exposed to pathogens that we may inhale. Normal, healthy lungs have a normal immune homeostasis. That is, there is a balance between the pro and anti-inflammatory processes. Our lungs have resident immune cells that can quickly respond to an immune insult, the pro-inflammatory part. Our lungs also have immune cells that can reduce inflammation or prevent an overreaction, the anti-inflammatory part. A disruption in this balance can be devastating, such as the cytokine storms in severe COVID cases. Now, let's talk about pesticides. At very high levels, pesticides can lead to death. However, most exposure occurs at much lower levels. Epidemiological studies have linked pesticide exposure to the development of heart disease, diabetes, asthma, and COPD. The pesticide I study, chlorpyrifos, was banned for residential use due to concerns of neurotoxicity in children, but it is still used in agriculture. I study the ability of low-level chlorpyrifos to disrupt the lung immune system. I measure the levels of pro-inflammatory markers, the percentages of immune cells, and signaling molecules that can enhance or attenuate our immune responses. I have identified an enzyme inhibited in the mouse lungs by chlorpyrifos that is identical to one in human macrophages. Macrophages being one of the most important immune cell types in the lung. I have also identified slight alterations in how our lung responds to an immune insult after chlorpyrifos exposure. I have identified a disruption in this immune balance between pro and anti-inflammatory processes. Thus, my research suggests that low-level pesticides can alter our lungs' immune response. This could be concerning for individuals living in agricultural communities during a pandemic such as COVID-19.
That concludes the presentations of the 8 3MT finalists. While our judges are establishing the grand champion and the runner-up, it's up to you, the viewers, to vote for your favorite presentation. Please visit the link in the chat to vote for your pick of the People's Choice Award winner. Thank you for joining us today for the 2020 Virtual 3MT Competition Finals. Please tune in tomorrow to find out who the winners are. Have a great afternoon.